Welcome everyone to the 33rd annual commencement for the Skaggs Graduate School of Chemical and Biological Sciences. I'm Keary Engle, and I have the honor of serving as this program's dean. It's a privilege to stand before you today, not only as a leader of the grad program, but as someone who deeply admires the resilience, courage, and determination of this graduating class in particular. This event is a special treat for me as an alum of this program, and this should also serve as a warning to this year's graduates that if you don't play your cards right, you might find yourself up here like me on the other side of this show in a few years. So this is my first year as dean. I took the baton from my distinguished colleague, Professor Phil Dawson, this past summer. And Phil leaves really big shoes to fill, pun intended. And I want to say a special thanks to Phil because all of these students benefited tremendously from his leadership over the past 7+ years. And he played a particularly important role in today's event because he lent me his fountain pen and bottle of ink so I could actually sign the diplomas. So next time you see him, say thanks for that. A little bit about the Skaggs graduate program for the friends and family in the audience. The Skaggs Graduate School has been ranked in the top 10 in chemistry and biology for the past 2 decades. We've got a host of dedicated faculty who are among the world's leading experts in their particular topics of specialization. And we have a unique model that is unlike traditional universities and empowers doctoral students to place research and the advancement of human knowledge at the forefront of everything they do from the day they first arrive on campus. Also, this year is a special milestone as we celebrated the 1,000th successful defended thesis from the program, which is an incredible milestone. And also the 1,001st, but that one didn't get as much of a fuss made about it. Each of these aspects on its own is noteworthy, but when combined with a dynamic environment of collaboration and synergy, they elevate Scripps Research to the position it holds today as one of the world's leading independent research institutes. Simply put, we do our darndest to inspire and train the next generation of scientific leaders, ensuring that the continued trajectory of scientific excellence is on the right path for generations to come. We're fortunate to have some of the best and brightest faculty training this next generation, and as you'll hear shortly in their personalized remarks, each of our advisors are deeply invested in the success of our students, showing tireless dedication, encouraging insatiable curiosity, and providing a collaborative environment with the freedom to pursue their research. Graduate students in this program continue to make vital contributions from treatments for several types of cancer and increasing longevity to mapping how the human brain operates and the intricacies of chronic diseases. These students have expanded what we know and reimagined what is possible. Today we have the honor of celebrating 61 tremendously talented students who've completed their degrees. 52 are here with me on stage today, and I don't know if we could fit many more. We've already seen their impact resonate across the entire scientific community. They've been cited in the peer-reviewed literature over 9,000 times and published collectively more than 220 articles. Every scientist here, yeah, we can give them a round of applause for that. As my father-in-law would say, not bad. Every scientist knows that these types of world-changing discoveries do not happen in a vacuum. And I want to extend a heartfelt thank you to the family members, friends, mentors, and supporters who are here with us today and those who are also here with us joining from around the world over Zoom. Your belief in these students, your countless sacrifices, your words of encouragement during long nights in the lab and hard quarters in the classroom, these things matter. And we know that today is your celebration too. So I want to ask the students to give a round of applause to their supporters who helped them along this journey. I also want to acknowledge the generous philanthropic community whose support has made opportunities, scholarships, and vital resources possible for so many of our students. Your investment has changed lives, and today is proof of that. Members of this class have received 28 awards and endowed fellowships, which you'll see named on the screen during the Conferral of Degrees. They were created by Scripps Research board members, faculty, and individuals and foundations passionate about scientific education, several of whom join us today. More than ever, philanthropic support in the face of proposed funding cuts and unprecedented disruptions to the research ecosystem will continue to sustain and bolster the opportunities for incredible students like these to pursue science and change the world. Graduates, you've walked a path that was anything but predictable. You experienced your graduate research and degree progression during a global pandemic. You adapted in the face of uncertainty, not just in your education, but to shifts in the world around you. That uncertainty continues today as we face the unprecedented challenges to the future of science and reduction of funding. Your persistence and strength has not only shaped your education, it has shaped your character. This perseverance will serve you far beyond these lab benches and classrooms. Today we celebrate your remarkable spirit of tenacity and what we know will become its transformative impact on the world. The diploma that you'll receive shortly is not just a symbol of academic achievement, it's a testament to your grit, your adaptability, and your refusal to give up when it would have been easier to tap out. You're entering the world not only armed with knowledge, but with resilience. And that, perhaps more than anything, is what the current landscape of the world demands. A round of applause for the class of 2025. Now it's my pleasure to introduce the President and CEO of Scripps Research, Pete Schultz.
Thanks, Keary, and welcome to the graduating class, their families, board members, faculty, and guests. We really are in interesting times right now in science. I think this class is better equipped than any class in the history of science with the tools and knowledge to have an impact not only on science, but on society. And we have great expectations. At the same time, scientific institutions, science itself, has a real threat to its sustainability and even the existence of major research institutions. And that's in part because I think the values almost everyone here holds, the value of science to society, the value of fact-based decision-making, and the belief that you should help those who are not as well off as you. That those values, I think, are being challenged right now. And the question is, what should we do? And the answer is, we certainly shouldn't retreat, okay? We should charge forward, because I think this institute has a history of taking on adversity and taking it on by being innovative and taking risk and doing things before the rest of the world realizes that's what should be done. And so that's what we need to do. And I used to use a movie analogy that Phil Barron liked, and that was The Last Crusade, okay? Where at the end of the movie, Indiana Jones has to get across the chasm to get to the Holy Grail to save his father. And I'd use the analogy that if you really do take on adversity and you just have confidence in yourself, a bridge would materialize over the chasm and you just walk across it, okay? I think now the better movie analogy is Apollo 13. Okay. We have to get back to Earth. And I think, what it's going to take is the kind of ingenuity and creativity and brain power that we've applied to our research. And I would argue, if we could understand the sense of touch and how to selectively activate a CH bond or regenerate heart, we sure should be able to find solutions to the challenges ahead. And that's what we're doing, okay? And the answer is you need to start thinking as an institution about controlling your own destiny, okay? For many, many years, we've depended on federal funding and philanthropy to do everything we want to do. And I would argue that we need to change that thinking, and we need to start thinking about a new way to support our science based on our own successes, and that's what we're doing. The fundamental strength of Scripps is basic research, and that's what we've been good at for many years. But now what we need to do is overlay on that basic research translational research. And what that will do is turn those discoveries into new medicines that will actually give back a very understandable return on investment to the taxpayers who fund all the science we do. But it also will create a whole new way to fund our science and reinvest in our own successes, and that's because as we move molecules further through clinical development into human trials, their value goes up, and if we do that ourselves, we capture that value and reinvest it in the scientific enterprise. And that's the model we're building, and I think no one else is building it like we're doing. And already, I would say over the last 4 or 5 years, we've moved 13 molecules into human clinical trials. So we've made drugs from our discoveries, and we're actually showing they work in human patients, okay? And those range all the way from cancer to neurodegenerative disease to infectious disease and to regenerative medicine. And importantly, we've become good at this, that I think, we've gotten foundations and pharma to support us to the tune of $500 million to do this. And if we're successful and we partner those, the revenues we get back will be 10 to 20-fold what we've gotten back historically. And that will provide a basis for the long-term sustainability and growth of Scripps Research. So, when faced with adversity, just have confidence in ourselves that we can actually solve problems, be willing to take risk, and be willing to innovate. And I think that really is the hallmark of Scripps Research. And finally, I'd just like to remind the graduating class that it really is a privilege to do science. Through my early career, I wound up leaving school and working night shift in a foundry, pouring molten aluminum and shaking out sand castings, okay? And what that taught me was, the opportunity to do science is an amazing privilege for us. It's almost like, artists like Monet painting paintings and, Mozart making compositions. We have that privilege, but remember that you always have an obligation with a privilege, and that's ultimately to give back to those that are less fortunate than you. And I hope you take on that responsibility and burden. So with that, we'll start with the Conferral of Degrees. And I'd like to state that these candidates have completed all the requirements for the Doctor of Philosophy degree. With the recommendation of the faculty of the graduate program at Scripps Research, I'm pleased to present them to you for conferring of the degree. And we will proceed in the order indicated in the program, and Donna Blackman will introduce the first candidate. And I should just preface this by saying the best things in life can be described in the fewest number of words.
morning. Jinhan Yu has made significant contributions probing a fundamental property of life known as biological homochirality. The chemical building blocks of life are single-handed. Our proteins and enzymes are made up of left-handed amino acids, and only right-handed sugars form RNA and DNA. In a series of high-impact papers, Jinhan demonstrated how this key signature of life may have evolved from a prebiotic world. Her work helped to take a problem that was once deemed experimentally intractable to give us a variety of possible mechanisms towards explaining the origin of life. But Jinhan's work is not only of fundamental scientific importance, it also leads directly to new methods and increased efficiency in the production of modern pharmaceuticals. Outside her work at Scripps, Jinhan is an avid musician who enjoys opera and musicals. She looks forward to a career in pharmaceutical R&D. Please join me in congratulating Dr. Jinhan Yu.
Hi everyone, I'm Mike Bolong and I'm Associate Professor in the Chemistry Department, and I have the immense pleasure of introducing our next three graduates, three absolutely stellar individuals from the second crop of students that have matriculated from my group. While losing all three at once is a bittersweet moment for me, I count myself truly lucky to have been positively influenced by these individuals, all of whom have made material contributions to the drugs we are helping to put into the clinic, as well as made their own seminal mechanistic discoveries in the field of chemical biology. So first up is Dr. Maya Boulos. During her PhD work here, Maya defined several pharmacological mechanisms for targeting a cellular signaling system in animals called hippo. This signaling system is highly conserved all the ways from flies to man, and it regulates organ size and regenerative repair. Understanding how we might drug this system with small molecules is absolutely paramount to uncovering how we might be able to harness our body's own regenerative capacity, not only to stop diseases of aging, but to reverse them as well. Maya did a lot in my lab, but Maya's PhD work ultimately culminated in one of the most mind-bending mechanism of action projects that I have yet to encounter. Here, Maya found a ubiquitin-like protein called FUBI can be directly targeted with a small molecule to activate a regenerative response. She showed that this protein is covalently conjugated to hippo members, regulating their activity, defining an entirely new dogma for how this pathway is regulated, and creating an entire subfield on her own. While I was many times uncertain if we could actually pull this off, Maya persisted, undeterred, and with extreme tenacity, working to define this signaling pathway in a few short years. Truly an amazing thesis. Congratulations, Maya. Next up to the stage is Dr. Kayla Elaine Nooch. During her time here, Kayla worked on drugging the hippo pathway also, but instead of focusing on upstream signaling, she focused on how we might target the most downstream part of it, the YAPT transcriptional complex. She realized early on that this regenerative response is often co-opted by cancer cells as a resistance mechanism to the many emerging targeted therapies like EGFR and RAS inhibitors. From this, she conceived of the methodology and executed an entire drug discovery project on her own. Working seamlessly with the team at Caliber, she brought forward an oral, once-a-day, covalent inhibitor that targets the 4T paralogs to the state of IND-enabling readiness. She did not stop there. Working from hypothesis alone, she discovered a new potential resistance mechanism to this drug. Going even further still, she showed that she could flip this inhibitor class of drugs into activators of the response, discovering a TEAD1 selective agonist, changing the paradigm how we think about this class of proteins might be drugged, and empowering new avenues for regenerative organ repair. Truly a stunning repertoire of work for one individual to complete in 4 years. Congratulations, Kayla. Last of the three graduates, but certainly not least, is Dr. Tiger Yu. Tiger's PhD work was incredibly diverse in its scope. He asked the question of whether we could use high-throughput screening, which is typically thought of as a kind of run-of-the-mill technology for discovering new ligands, but to go beyond this, expanding the scope of tools that might be available for biologists and chemists alike. To this end, Tiger has been incredibly productive, delivering 4 first author works on his own. He single-handedly provided the first chemical tools to study 2 mammalian signaling pathways. Tiger also devised a screening platform and generated a computationally derived fluorophore library to identify new fluorogenic dyes. And lastly, he devised a technology platform for profiling large chemical libraries for their reactivity to engage cysteine, uncovering new allosteric activators. I will be lucky enough to keep working with Tiger. I got to hold on to one of these graduates at least, as Tiger has taken a position at Caliber, where he's continuing to push the limits of small molecule drug discovery, looking to see if we can expand human lifespan and health span with small molecule drugs. Great work, Tiger. Congratulations.
Hi everyone, my name's Dennis Burton. I'm the Chair of Immunology and Microbiology, and I have the honor of presenting 2 new PhDs today. The first is Nate Beutler. I'll just say that the last Dr. Beutler who was here at Scripps got a Nobel Prize, so expectations are high. Nate will actually only get 1 PhD, but he did 2 PhDs worth of work, one on COVID and the second on malaria. He began his graduate studies leading into the pandemic, and he quickly became one of the key members of our team that generated antibodies to SARS-CoV-2, the guilty virus. His work was to use live virus and develop test tube assays to see if our antibodies were any good. And they were very effective. Folks have probably forgotten now what it was like in March, April, May of 2020 and how concerned we were at the time. In fact, the team, including Nate, didn't allow me into the lab at all. I thought at the time that was very sweet and nurturing, but I later heard that they said, well, you know, if Burton shuffles off this mortal coil, we may have some employment issues. So. But the pandemic ebbed, and then Nate turned to malaria, which is his real love. It's sort of weird that scientists should love pathogens, but we do, believe me. And he made 2 highly significant contributions. He generated a whole series of antibodies to malaria, which we're using for vaccine design, and he set up an insectary, breeding mosquitoes, infecting them with malaria, and then rescuing the parasite from the salivary glands of the bugs, so we could study the form of the parasite that is injected into humans. And he's doing his work with Andrew Ward using cryo-EM and continuing his studies now at UCSD, and we're very excited about what this is going to teach us about malaria. So, congratulations, Dr. Beutler. So the next new doctor is Selena Keating. Her work was even more tied into the COVID pandemic than Nate's. Let me just remind you of the early history of the pandemic. Early December 2019, cluster of cases in Wuhan, China. Early January 2020, the cause was identified as the novel coronavirus, SARS-CoV-2. Mid-January, spread to 2020, spread to the US. Early March, a global pandemic declared. Late March, Scripps shut down, except for COVID work. And at that time, there were no vaccines, no effective drugs, And there was great interest in generating monoclonal antibodies, and I just described that work that Nate did. But we wanted to show that these antibodies actually worked in vivo, in animal models. And that's what Selena did, which was very brave at that time. She'd been working on animal models for flu, and she took the virus and looked to see how effective the antibodies were against SARS-CoV-2 in hamsters. And that was a really a superb piece of work, and the study came out very, very quickly. It's actually published in Science in June 2020. And I'll just remind you at that time, again, how bad the virus was at that time. The death toll in the US by the end of May 2020 was greater than 100,000. By the end of September, it was greater than 200,000. So understanding of antibodies, important for vaccines and so on, was very important. And Selena was absolutely key in that. And then she went on with Joe Dardeen to develop her skills in antibody engineering in a whole set of different experiments. She's now heading towards a career, I think, in business development. So, Dr. Keating, many congratulations.
Hello, I'm Holly Klein, and this is Anton Maximoff. We're both in the neuroscience department, and it's a pleasure to recognize Yi, who has done her thesis work mostly in my lab, but also in collaboration with Anton. So what Yi was interested in addressing was really fundamental information about how our brains develop and how we learn. And one of the key triggers of those events is that there is activity in our brain, for instance, when we hear a baby crying in the background. And as a result of that activity, new genes are transcribed in our neurons, and those genes generate new proteins, which are important for changing the circuitry within our brains. Now, even though we know a lot about that phenomenology, we don't actually know a lot about the nuts and bolts of changes that occur in our brains. And what Yi did amazingly was carry out an unbiased screen to identify newly synthesized proteins that are triggered in response to these events from which we learn. She did a remarkable series of experiments, ran into a bunch of roadblocks along the way, and every time she hit a roadblock, she creatively, thoughtfully found a way to solve those problems and move forward, and made amazing discoveries which have changed our understanding of brain plasticity and development. So thank you, Yi.
It's been a pleasure to be Yi's co-advisor. Dr. Klein just outlined her remarkable accomplishments, and I'm not going to repeat that. What I would like to add is that I believe that one of the most important goals a graduate student must achieve is becoming an independent thinker, and Yi has clearly excelled at that. I have also absolutely no doubt that she will succeed on whatever career path she chooses. Congratulations.
Good morning. I'm Assistant Professor Mike Constantinides, waiting on Adam. All right. So it was a pleasure to mentor Dr. Adam Sobel, who displayed an innate curiosity from the moment he joined my lab. This led Adam to ask how antibiotic use in early life impacts the immune system. And this was a question that was highly relevant to him because he welcomed a daughter during his graduate studies. Through a series of very challenging, long experiments, Adam determined that specific antibiotics can deplete commensal microbes that are beneficial for the development of the immune system. And this renders mice more susceptible to bacterial pneumonia. Not only did he identify an issue, but he also solved that problem by determining that certain bacteria can actually restore immunity if given during the time of antibiotics. And this leads to potential probiotic therapies that could be beneficial for neonates. The skills Adam learned and the perseverance he displayed during his PhD will serve him well, as he now performs high-throughput screens of small molecule protein interactions at JLab's startup just down the road. So congratulations on your achievement, Adam, and I look forward to seeing what you'll achieve during the next stage of your career. Please join me in congratulating Dr. Sobel.
Hello, I'm Ben Cravat from the Department of Chemistry, and it's my pleasure to introduce Wanjin Zheng. Wanjin. In our lab, Wanjin made major contributions to the fields of small molecule probe discovery and chemical proteomics, culminating in a recent first author paper published in Nature Chemical Biology. In this manuscript, Wanjin introduced an innovative paralog hopping strategy to expand our understanding of small molecule protein interactions in human cells, leading to the discovery of a cryptic druggable pocket in a highly cancer-relevant adaptor protein kinase complex called CCNE1-CDK2. There are now several oncology drugs in preclinical and clinical development targeting this novel site that Wanjin helped to discover. This project also involved a close collaboration with Pfizer scientists down the road, and Wanjin navigated this interaction with admirable maturity and collegiality. Wanjin's scientific accomplishments are matched by an adventurous spirit. She's an avid hiker and tackled many of San Diego's most difficult trails. She also loves Hawaii and actually gave our lab a pretty frightening scare in 2023 when she had to escape the Lahaina fires. She made it back well and has graduated and is now performing postdoctoral studies in Zhiyang Zhang's lab at UC Berkeley, where I'm confident she will continue to excel. Wanjin, congratulations. Please welcome me and thank you for wonderful accomplishments.
On behalf of my distinguished colleague, Ben Cravat, and co-mentor to Adrian Guerrero. So when I think of Adrian, I think of two words, tenacity and creativity. Adrian came to us from the University of Texas at Rio Grande, and he earned both a chemistry degree and a degree in biology, and applied to the UCSD MD-PhD program, and he came here to earn his PhD. And here, he worked on a protein that's critical in attenuating inflammation so that it doesn't become too aggressive, called alpha-1 antitrypsin. And he figured out a really nice pharmacologic strategy whereby you can use a combination of drugs to both enhance the biology as well as the chemistry of protein folding. And I think this strategy is going to turn out to be quite useful for ameliorating both COPD and related diseases. Adrian's parents and friends are here with us today, and Adrian, I'd like to congratulate you for a fantastic thesis, and we expect great things from you.
should have worn heels. All right, my name is my name is Laura Solz, and I'm the Associate Dean of Graduate Studies for the Florida campus. I'm pleased to introduce Samantha Meyer, whose advisor has recorded their remarks. It should be starting.
So hi, I'm Matt Disney, faculty member at Scripps Research on the Florida campus. Very grateful to be giving this video for all the 2025 graduates. So congratulations to all of you guys. It's really a testament to the program for all the successes, all the science that you produced, and I'm really grateful, as are all the faculty, to have seen you guys grow from first-year students to now fully minted PhDs. And so thank you for contributing to the culture, the excellence in research and science that we do. I mean, science is very difficult, and you really have to be focused to be able to achieve getting a PhD, earning a PhD from Scripps, but also contributing to the broader scientific ecosystem, which unfortunately we're all going to have to deal with, is through some choppy waters, just like this painting behind me. And so you got to be partners on smoothing these out. Unfortunately, I can't be with you today because my son's graduating. And so I want to be especially grateful, Sam, to you. Samantha Meyer is a student in our lab that's graduating. Very grateful for you, Sam, for all the work that you've done in the lab. Very proud to see you develop into the scientist that you are today. You know, you've done some pretty amazing things in the lab that I think a lot of people didn't think were possible to do, but with diligent hard work and some good ideas, you were able to pull it off. And so I'm really looking forward to the future science that you're going to produce in industry, and really to fulfilling a lot of the promise that the science has to deliver medicines to patients. So, you know, again, I want to just take the time to be grateful for all of you guys, and the program in general, you know, that we've here contributed to and are very proud of also. And so congratulations, everybody, but especially Sam and your family. And so good luck in the future, and I'm sure we'll stay in touch. So thank you.
Good morning. It is my pleasure today to introduce Dr. Yin Wu. Yin came to Scripps after graduating from the University of Science and Technology of China, where she obtained a Bachelor of Science, Life Sciences degree, and she was in a special class for the gifted young. Yin joined my lab, and from the get-go exhibited two defining traits that I believe are critical indicators of success. The first one is grit. If you look up grit, the definition of grit is courage and resolve, or strength of character, and Yin has grit. Yin set up a brand new system in our lab where she set up to study a model for Alzheimer's disease in neurons in a related group of neurodegenerative dementias called tauopathies, and these include frontotemporal dementia. This system had not been characterized previously, and it was not an easy task. She encountered various stages of optimization and troubleshooting, which she had to surpass and did so brilliantly. With this system, Yin unveiled a brand new cellular pathway in neurons that accounts for the formation of these pathological lesions that are very common in characteristics observed in humans with Alzheimer's disease. And with this, she unveiled and opened new pathways for the test and testing of therapeutics that are really important and new for these diseases. The second trait that Yin exhibits is that she loved to question things. Yin is very a deep thinker and an avid reader of literature, and she's not afraid to voice her disagreements or dissents about concepts, ideas, opinions, and experimental approaches, especially those from her advisor. And this is keenly important, and a very important trait, I think, for scientists, and is most all that an advisor can hope for. So in addition, Yin is gifted at the bench. She possesses a green thumb and is a Ferrari when it comes to molecular biology. Yin is also a wonderful lab citizen, and she has an admirable work ethic. She's one of the hardest working scientists I know. As a result of her work, Yin has contributed significantly to our knowledge of tauopathies. She's an author on 6 publications from her tenure here at Scripps, including some with collaborations that she forged herself. So these qualities will serve Yin very well in her new position as a postdoctoral fellow, who she's securing now. And it's been a really true pleasure, Yin, to have an opportunity to work alongside you. I can't wait to see the great science that will come from your postdoctoral studies and beyond. Congratulations.
It's me again. It's an absolute pleasure to introduce Dr. Omar Apolinar, or more precisely, Dr. Omar Apolinar, who graduates today with a PhD in chemistry from Scripps Research and a DPhil in biochemistry from the University of Oxford as part of our Skaggs Oxford Scholarship Program, while publishing 12 papers across these 2 experiences. I first came to know Omar back in 2018 when he was an undergraduate student at CSU San Marcos and joined our highly selective summer undergraduate research fellowship program. Omar made a big impression, contributing to a co-authored publication as an undergrad. We were then delighted to be able to recruit him back to Scripps Research, where he earned an NSF graduate research fellowship upon matriculation. In my lab, Omar achieved something I thought may be impossible, though I didn't tell him that until now, so surprise, which was taking a Nobel Prize-winning reaction called the Suzuki cross-coupling, widely used in assembling the core carbogenic skeletons of medicines, and making it better. In particular, he developed a 3-component version of the Suzuki coupling that integrates a carbon-carbon double bond and discovered an earth-abundant nickel catalyst to make the reaction enantioselective. In other words, as we heard from Professor Blackman, selectively producing only the right hand of the molecule over its left hand so that it can give a proper handshake to the protein target of interest. At Oxford, he continued this breathtaking trajectory by developing a fundamental understanding of the coordination chemistry involved in taking fluorospar, a bulk mineral, and converting it into valuable organofluorine compounds like the Teflon coating found in your pots and pans. Since graduating, Omar has taken a position working in the area of AI with Maycor and has an eye on a career in drug discovery. Congrats, Dr. Apolinar. Turning now to the second graduate from my group, Dr. Camille Rubel is a native of Agoura Hills, California, and as an undergrad attended the storied College of Chemistry at UC Berkeley, where she performed research under the mentorship of an organometallic legend, Professor John Hartwig. Camille first came onto my radar during a seminar visit that I took to UC Berkeley, where my host, John Hartwig, arranged for me to meet with 2 of his outstanding students. After being riveted by their tales of iridium-catalyzed CH silylation, I was shocked to learn that one of the students, who happened to be named Camille, was not a PhD student, as I had assumed, but actually an undergraduate who happened to be applying to graduate school that very same year. How convenient, I thought. I strongly encouraged her to consider the lab of an up-and-coming, though sometimes cantankerous, researcher, yours truly. And the rest, as they say, is history. During her PhD, Camille proved to be a force of tremendous positive impact both in and outside of the lab. Her graduate research took her on an adventure that included a stint in Lyon, France, and collaborations with groups at Georgia Tech, Wisconsin, Pittsburgh, and Bristol-Myers Squibb, all under the auspices of democratizing nickel catalysis, developing practically convenient catalysts and procedures that can be deployed anywhere in the world without specialized equipment, like clunky inert atmospheric glove boxes, thereby lowering the barrier for adoption of this earth-abundant metal with profound potential to replace the precious metal catalysts that we rely on, like palladium and platinum, that are vanishingly rare on Earth's crust. Her discoveries have already been recognized by nationally competitive awards, including the Kenneth Hancock Award by the American Chemical Society and the Merck WCC Research Award. Outside of the lab, she was a beacon of positivity during the COVID-19 pandemic, a beloved teaching assistant for the organometallic chemistry class that's taught here at Scripps Research, and a community leader who expanded Scripps Research's partnership with the Barrio Logan College Institute, which provides mentorship and programming for disadvantaged youth in downtown San Diego. Camille's PhD culminated in 12 publications, and at present she's a postdoc at MIT with the physical electrochemist, Professor Yogi Surendranath. Congrats, Dr. Rubel.
Hi, me again. You're going to be sick of me by the end of this presentation. Okay, so I am pleased to introduce Andy Pan, whose advisor, Mike Farzan, has recorded their remarks.
Hi, everyone. Sorry I couldn't be there, but I couldn't be prouder of Andy and the body of work that he accomplished in my lab. Andy started by mastering a very complicated technology and then showed his mastery of that technology by pushing it in totally new directions and inventing by himself a new variant of the same basic approach. So the first part of his thesis, which he published in Nature Biomedical Engineering, shows that you can edit primary B cells to express essentially any protein therapeutic and affinity mature that protein therapeutic in a mouse to make them more potent and bioavailable. The second part of his thesis describes his original invention, which he calls fusion BCR, which now transforms that technology, opening up new directions for it and allowing us to address new questions in B-cell biology. His creativity here has been matched by a consistency and rigor of his work, and it's just been a pleasure to mentor him scientifically. But I also would like to mention something that I think might even be more impressive. As you know, the years covering Andy's PhD have been a challenge with the pandemic and the Florida transition and changes in administration that have been especially hard for our international students. In Andy's case and with his peers, they were told initially that they could not move with their mentors to a new institution. Andy led our effort to challenge this, ultimately giving all of my students the option to move with me to Harvard. In the process, he showed real guts, combined with his trademark level-headedness and calmness, as well as some selflessness, since his own visa situation ultimately allowed him to move regardless of what happened to his peers. So I think it's especially important to appreciate someone who has shown, as Andy has, the kind of ethical backbone that would be so welcome now. Today, though, is about his scientific accomplishments. I want to congratulate Andy for his outstanding work and his parents for raising him so well. Thanks.
Good morning, everyone. I'm Stefano Forli. I'm in the Department of Integrative Structure and Computational Biology, and today I have the privilege of introducing two brilliant graduate students that graduated from the lab. The first one being Althea. So, as I repeated multiple times in several occasions, it was really an honor and a privilege to mentor you. You are one of the students that started during the pandemic. You did the rotation entirely virtual, and I was pretty sure that we succeeded at scaring you away. It didn't work out, and you decided to join our lab. So it was easy for us because when you started, you were already a very accomplished, an exceptional MD PhD student, and so Still, it was very fantastic to see you growing to become the scientist that you've become today. And since then, you've been a beacon of integrity and dedication for your colleagues, with boundless curiosity and nearly endless energy, to the point in which, and I apologize on behalf of her to all the other computational graduate students, you crashed the Scripps supercomputers multiple times, eager to get your calculations done. Well done. So I'm sure that your numerous achievements, minus the crashes, will inspire future generations of the students in our lab and also in the program. And you made me a very proud mentor. So congratulations for your very well-deserved PhD. Okay, the second graduate student, Batu, came all the way from Mongolia before a quick stop in Japan, and he decided that getting one mentor was too easy, so he decided to get two. So I had the pleasure to commentor him with Andrew, and so you decided to take a challenge on mastering very complementary but different disciplines like structural methods and computational methods. So you decided to tackle another very difficult target, which is autopetry. It's a protein that is involved in the perception of equilibrium in our inner ear, among other things. So the target was difficult, but you persevered until you succeeded. And through this journey, you developed a very broad skill set in biochemistry, structural biology, computational biology, and you should be very proud of this because you were very successful despite all the adversities. And so I know that there's some of your family that traveled all the way back from Mongolia to visit you, and I think, they deserve to hear it. Tahun hir on zoim, so you're truly exceptional.
Hello, everyone. My name is Danielle Grochan, and I'm an assistant professor in the Structural and Computational Biology Department. And it's an honor to introduce Dr. Michaela Medina, the first graduate student in my lab. During her graduate studies, Michaela became one of the first and few experts in the world in a cutting-edge imaging technology called cryo-electron tomography. Now, this is really the stuff of transforming what we thought was scientific fiction into reality, because this approach enables us to look at protein structure inside the cell without having to do any purification. And Michaela has really spearheaded the efforts here at Scripps, and anyone who's ever heard of tomography at Scripps or has benefited from the infrastructure really has Michaela to thank for setting this all up. She focused this expertise on studying mitochondrial structure, which has resulted in 4 publications, one of which she earned co-first authorship in the Journal of Cell Biology, which has now been cited nearly 50 times. She's also been recognized numerous times with awards and fellowships, including a fellowship from the ARCS Foundation, as well as the Student Research Achievement Award from the Biophysical Society. She's presented her work many places, including already serving as keynote speaker at the Inspire Symposium, as well as presenting her work internationally at an EMBO workshop in Heidelberg, Germany. Michaela, your light and drive and kindness has been felt by so many in the lab. Thank you for really shaping me as a mentor, teaching me everything I need to know about K-pop, and for really inspiring many generations to come. Congratulations.
I'm Scott Hansen. I was previously on the Florida campus. I've now moved my lab to the Chinese Institutes for Medical Research in China, and it's my pleasure to introduce Zixuan Yuan. Zixuan was born in Changsha, China, to Keary Engle and Wangling Yuan, and I want to commend them. They raised a very good person and a bright scientist, and the apple doesn't fall far from the tree. In 2019, she joined the Scripps Research program and my lab with the anticipation of studying sleep and anesthesia. But like many of her class, that was interrupted by COVID. And my lab knew nothing about virology. But when I asked her, well, do we want to go home or do we want to continue working? And the only option was to study COVID. She agreed to study COVID. And in June of 2020, had deposited her first author paper in bioRxiv, and later that was published and was downloaded 160,000 times and has been cited over 60 times in the last 2 years. And it's remarkable coming from, that is from a graduate student, not from the lab, to learn the virology that was required for that paper. But then, of course, she returned to her what she really wanted to study, and just last month published a second paper on GABA and activation by a positional shift in the membrane. And it's a completely new way of thinking about how a neurotransmitter affects an ion channel, and I have no doubt in the future will change the way we think about neurotransmission. Zixuan has many strengths. I will just highlight two. One is the ability to learn. She's humble, hardworking, and open-minded, and that ability, those skills are important for being able to learn. And the second one that I didn't appreciate until I saw it at work in my lab is that of networking. Many scientists do not pursue social interactions, and she's actually very good at it, and I think that it will come in very handy as she moves forward. She's accepted a position in the Nettergard lab in Rochester, which is a very competitive lab where she'll be able to continue studying sleep. And she also has influenced my lab substantially. When I was thinking about moving, she knew the people that had started the institute that I'm now at and gave me the confidence to move to China, and probably has affected my life about as much as anybody. I want to say in Chinese, Ju Yuanbo Shi Wei Lai Ifan Fen Shun. Congratulations, Dr. Shun.
Good morning, everyone. My name is Mia Huang. I am a professor in the Department of Chemistry, and I'm pleased to present Meg Critcher to you today. Meg Critcher obtained her bachelor's degree in biology from the University of Bath in the UK. During her time at Scripps, Meg has contributed to 6 published papers, 5 of which list her as the first or co-first author, one of which is the flagship publication in my lab in Nature Chemical Biology. Meg is a true testament to the interdisciplinary nature of our graduate program here at Scripps. She came to my lab as a fly biologist, and she's now graduating as a seasoned chemical biologist. Her dissertation is entitled Development of Chemical Biology Tools to Probe Proteoglycan Structure-Function Relationships, and her expertise in proteoglycan biology and for making excellent figures is unmatched in the lab. Congratulations, Dr. Critcher.
Hi, my name's David Namasi. It's a pleasure to introduce a graduate student who worked with me, Linghang Pan, who I hope we see quickly. Hi, Linghang. Good to see you. Actually, Linghang preferred to work a bit later in the day, so we're used to him coming a little bit later than we expect. But that started during COVID. So Linghang is from Wuhan, and sort of early on, he was quite interested in the virus, and so he sort of diverted from his original project for a time to help a number of labs at Scripps better understand how to neutralize the virus, making pseudoviruses, and with every variant of concern, he updated the system. But towards the end of his career, he got back to his original project, which was working on phospholipase D family nucleases in the lab, and completed a series of studies that helped us understand better how to perhaps inhibit these enzymes for certain applications, but also to understand variants that are related to diseases like Alzheimer's and systemic lupus. So Linghang, thank you for joining my lab. We had a wonderful time with each other, and thank you for being such a good friend to many at Scripps.
Hello, I'm pleased to introduce Roberto Salatino, whose advisor, Michalina Januszewska, has recorded her remarks.
Hello, my name is Michalina Januszewska, and it is my pleasure to introduce Roberto Salatino. Roberto received his BA in Mechanical Engineering from Bari University in Italy and BSc in Chemistry with Biology emphasis from University of Utah. Subsequently, he worked as a lab technician at the Center for Clinical and Translational Sciences at the University of Utah, and in 2015, he joined Tavazzoli Lab at Columbia to work on new methods for large-scale proteomics. He joined my lab in 2019 as the first graduate student of the lab, and he undertook 2 massive projects. In the first project, he spent long hours optimizing the protocol for our new method that allows for cell sorting based on their mutational status and subsequent bulk and single-cell sequencing or chromatin profiling. As his second project, Roberto took to develop primary cell lines from multi-region biopsies from brain tumors. His patients in that matter yielded over 35 cell lines and at times 4 incubators of capricious neurospheres. And this massive amount of work will allow us to move forward with drug screening capturing intratumor heterogeneity for those most aggressive brain tumors. Roberto, congratulations on your graduation and all the best wishes for your future endeavors.
I'd like to welcome Carolyn Coco to the stage. I'm Jeff Kelly in the Department of Chemistry, and Carolyn graduated from the Scripps Oxford program, also funded by the Skaggs family, and so she gets a PhD at Scripps and a DPhil at Oxford. So Carolyn's journey here started out through a phenotypic screen that we had the privilege of doing with Caliper, where we are looking for small molecules that hasten lipid droplet clearance in the quest of trying to find autophagy activators. And Carolyn realized really quickly that the molecule she started studying wasn't an autophagy activator, but instead did something incredibly interesting, which had activated the integrated stress response through a target and a mechanism that had never been described before. So I think it's fair to say that in Carolyn's PhD work here, that she discovered a new small molecule that activated a novel pathway through a novel target. And that pathway is pretty important in degenerative diseases, and hopefully someday that will be used in humans. So Carolyn, it's not an understatement to say that you've changed the world, and we expect great things from you. So I've also had the privilege of mentoring Sergio Labra. So Sergio truly earned his PhD. He came to us from the University of Pennsylvania, where he got a degree in chemical engineering and also a master's degree in biotechnology. And what we set out to do here was to take advantage of the award-winning work of Yamanaka, where he showed that you could differentiate stem cells into 2D cultures. And with Stuart Lipton's lab, we made three-dimensional cultures called organoids, which are effectively mini brains. They have pretty much everything except the components of the immune system called microglial cells. And Sergio was able to show that, in fact, even in these early models of brain development, we already see the degenerative features of Alzheimer's disease in hereditary AD-derived iPSC cells. And moreover, he was able to use these cells to identify new mechanisms of action that might be useful in ameliorating pathology in Alzheimer's disease. So, and he had some serious setbacks along the way that he pushed through. So he earned his degree in every respect of the word. Sergio, we expect awesome things from you in the future. We welcome your family here as well. Last but certainly not least, I'd like to introduce Karina Negroho. So Karina came to the Scripps graduate program from earning a biochemistry degree at UCLA. And Karina, I would say, is fascinated with proteomics, chemoproteomics, a field that my colleague Ben Curvat largely invented. And so what Karina did for her PhD thesis was to make molecules of intermediate complexity harboring weak electrophiles, and then she screened them against living cells to discern whether there was any proteins in the cells that reacted with those small molecules. And she found a very important target in the degradation of ER proteins, and is probably an inhibitor of ER phagy. And Ben's lab had the privilege of discovering a similar molecule. And hopefully, owing to Karina's discovery, we can actually figure out how to use this in a clinically meaningful way. I would also say that in collaboration with Gabe Lander's lab, Karina enabled a study that showed that the structure of transthyretin that we had been banking on for almost 30 years really wasn't right in terms of what exists in solution. And so it explained why we had to make so many molecules to ultimately end up stabilizing the protein. So I think she did some really nice things in her PhD and truly earned it. Congratulations, Karina.
I'm obviously not Doug Coteen. Unfortunately, he couldn't be here today. Something unexpected came up, but I am pleased to introduce his student, Chris Williams. Chris grew up in New York and attended Hamilton College in New York, graduating with a BA in chemistry. Prior to attending Scripps in August of 2019, he worked in 2 post-baccalaureate positions. First as a lab tech in Evonik Industries, and second as a medicinal chemistry scientist at the MD Anderson Cancer Center. Chris's background in chemistry was particularly useful for the project he has worked on while pursuing his PhD in the Coteen lab. Understanding the mechanism by which the prolyl isomerase enzyme PIN1 influences the structure and function of the nuclear receptor transcription factor PPAR-gamma. Chris's studies led the Coteen lab into some unanticipated directions, both in terms of not anticipating how complex his studies would be, but also in terms of the potential impact on the field. Chris's work, which is scheduled to be published on the PNAS website next week, identifies a tethering mechanism that underlies PIN1's enzymatic modification of PPAR-gamma, the first such non-classical mechanism to be reported in the PIN1 field. During his time in the Coteen lab, Chris was awarded an NIH Ruth L. Kirstenstein Predoctoral Individual National Research Service Award, that's a mouthful, F31, for his studies on PIN1 and PPAR-gamma. Chris's path during his graduate studies has been nonlinear, including a move to Nashville, Vanderbilt, 2 years ago to finish his graduate studies in the Coteen lab. Your tenacity and ability to stay focused and motivated during tough times is admirable. Life after Scripps for Chris will hopefully include a transition to industry, and Chris currently has some promising opportunities. Congratulations, Chris, on a job well done.
Hello, my name is Luke Wiseman, and I am delighted to be introducing Mildred Kissay on behalf of her advisor, Luke Larson, who was unfortunately unable to be there today. Millie had a unique path getting to this stage today. She was born in Dar es Salaam, Tanzania, and lived there until she was 11, when she then moved with her family to Doha, Qatar, where she attended middle and high school. Millie then moved to the US to attend college and obtain her BS with a double major in chemistry and biology from Bard College in New York, and prior to coming to graduate school, Millie worked for 3 years at Moderna. At Scripps, Millie's PhD studies focused on applying her skills and passion for protein science to characterize small molecules identified from cell-based phenotypic screens. Her primary work, done in collaboration with the Wilson Lab, was published in Bioorganic and Medicinal Chemistry Letters, and she graduated as a co-author on a paper published in Science. Today, Millie is joined by her mother and sister. On behalf of both Luke's and the graduate school, Millie, congratulations. I am also happy to introduce Philip Sander, again on behalf of Professor Larson, who is traveling. Philip grew up in Bonn, Germany, and received his Bachelor's of Science in Molecular Medicine from the University of Bonn. Prior to coming to Scripps, he obtained a master's degree in biochemistry and biophysics from the University of Freiburg, during which time he also worked as a visiting scholar at Berkeley developing RNA-based oligomers. For his PhD work here at Scripps, he ended up working primarily on a project that was fairly different from most of the research that is done in the Larson lab. Specifically, he worked to develop a generalizable approach to perform phenotypic screens of mammalian cells using DNA-encoded small molecule libraries. His primary work was published in RSC Chemical Biology, and he also graduated as a co-author on papers published in PNAS, BMCL, Cell Chemical Biology, and Science. Today, Philip is joined by his daughter Charlotte, his wife Katerina, and his mother Carol. On behalf of Luke and the graduate school, Phillips, congratulations.
Congratulations.
It's my great pleasure today to introduce to you Rebecca Mello, who, who recently completed her PhD work in my lab. So my lab studies circadian biology, and as you may or may not know, shift workers and others who are subjected to disruption of their circadian rhythms are at greater risk of many diseases, including cancer. And Rebecca's PhD thesis work focused on this, trying to understand this intersection between circadian disruption and cancer biology, with a particular focus on the most common type of kidney cancer, clear cell renal cell carcinoma. Rebecca's work focused on an interaction between the circadian transcription factor BMAL1 and the hypoxia-inducible factor HIF2 alpha that plays a key role in driving clear cell renal cell carcinoma. Building on Rebecca's findings, she made a striking discovery that the efficacy of a drug called belzutifan that was recently approved for treating renal cell carcinoma, the efficacy actually is dramatically different depending on what time of day the drug is given. And we're now partnering with renal oncologists at Scripps Health to follow up on this great work that Rebecca has done. Her impact in the lab went far beyond her scientific discoveries. She's just been a fantastic team member. She's been a mentor to many other students, and she has forged collaborations with experts in the field, including Celeste Simon. And I'm going to miss her greatly when she leaves, which I hope won't be too soon. So please join me in congratulating Rebecca again.
Hello and welcome everyone. I'm Chris Parker. I'm a professor in the Department of Chemistry, and today is my great honor to introduce Dr. Ines Forrest and also to welcome her family and friends who joined us from all over the world today. Part of a military family, Ines was born in Lorraine, France, and then completed her undergraduate studies at ECPM in France. Then she would subsequently receive not only just one, but 2 master's degrees in chemistry and chemical engineering. She wanted a 4th degree, apparently, so she joined my lab as my 3rd graduate student. There she worked, her thesis work mostly focused on expanding the boundaries of targeted protein degradation, which uses small molecules to redirect disease proteins in the cell to the cell's trash can for their elimination. This method had previously been limited to a small subset of proteins, and so Ines, working collaboratively with AbbVie, developed a powerful strategy to globally and agnostically identify proteins that can be degraded by small molecules, significantly expanding the scope of this therapeutic modality. And so this had culminated in patents. We were able to even acquire some seed funding from venture capital firms, a review, and an exciting preprint that is currently under review at a top journal. Fingers crossed on that. In addition to tearing it up in the lab, she also racked up a ton of accolades, including the very prestigious Ford Foundation Fellowship, the CAS Future Leader Award, the ACS Graduate Student and Postdoctoral Scholars Recognition for Leadership Award, and many others. Ines is what I would call an all-terrain scientist. She can build molecules. She's mastered proteomics. She's also an expert in molecular and cellular biology. She really could do anything. On top of that, she's brilliant, absolutely resilient, and hardworking, and it's been a great pleasure hosting her. Ines, congratulations, super proud, and very excited to see what your future holds.
Hello, everyone. I'm Jim Paulson, Department of Immunology and Microbiology. And it's my great honor to introduce two students to you today. The first is Brett Garabedian. Brett got his bachelor's degree at UC Berkeley and was in the process of getting his master's degree at the University of Basel when he received word that he was accepted into the graduate division. And I happened to be giving a lecture in Basel at the time. And so he arranged a breakfast meeting and arrived with his future wife, Gosia, and pitched an idea to me for a thesis project. Well, it was a great idea, and soon thereafter he joined the lab. So Brett is a voracious reader of the literature and is always full of ideas, both for his own research, things he wants to work on, and also the research of others in the lab that have benefited from his thoughts. Several of these ideas led to his own thesis work. He developed novel strategies to enhance the immune system's ability to attack cancer by disrupting the glycan shield that cancers use to prevent immune system attack. This has been the result of two outstanding papers that are in late revision. But it doesn't stop there. Another idea he had was the basis for his oral research proposal, which is a requirement by the graduate division. He liked it so much, he wanted to keep working on it as a side project, and it has resulted in the generation of a startup that is now funded with seed funding. And in fact, in about a month from now, he will join that full time to lead that company forward. So outstanding, really. And I look forward, first let me just say that he's joined today with his wife, Gosia, and their son, Artie, and his wife, his mother, Bonnie, and oldest brother, Grant. So congratulations, Grant. I look forward to following your progress. Yes, please. And now we can go. And the next candidate is Eleanor Bashan, and I'm joined by Peng Wu, Professor Peng Wu. We were joint mentors of Ellie. So Ellie received her BS at Ohio State University and joined the TSRI grad program there shortly after. She approached us because she wanted to pursue a project that embraced the common interests of both the Paulson and the Wu labs. She was, and Peng will talk more about that in just a moment. She was considered a full member of each laboratory. I really don't know how she managed it, but she kept us so well informed that we rarely had to talk to each other about her research project. In other words, she managed us very well. Ellie always made time to help others in the lab, and she was the one in my lab that was responsible for making celebrations for people's birthdays and making sure everybody signs the card and that kind of thing. While she receives her PhD today, she'll be entering medical school at Ohio State University, and she has a desire to combine both her PhD and an MD so that she can do research that will be translated directly to patients, which I think is an admirable goal. She's joined today by her family, her mom and dad, Jack and Veronica, and her brother, Sam. Let me turn it over to Peng.
So Ellie's thesis work was focused on the glycoimmune checkpoints cyclic 7 and 9 as their mouse homolog cyclic E. The interaction of these inhibitory receptors with sialylated glycans resembles the interaction of PD-1 and PD-L1, recruiting shift phosphatases to the immunological synapse, where they trigger immune suppression. Ellie discovered that selective removal of cyclic E ligand where CRISPR-mediated knockout of the enzyme responsible for the creation of cyclic E ligand on CD8 T cells can significantly enhance their anti-tumor immunity in multiple mouse solid tumor models. Mechanistically, this enhanced tumor control requires cyclic E expression on macrophages and is driven by the improved CD8 T cell differentiation, tumor infiltration, and cytotoxicity. In the past 5 years, Eli has published several papers, one co-first author paper in JACS, and we plan to submit her manuscript on Skaggs very soon. So Eli has done a lot of work for the lab, serving as a safety officer, and she also helped to create a new lab website for us after Jay Sharpless pointed out to me that our website was outdated. Eli, we are very proud of you. Congratulations.
On behalf of Matthew Pipkin, it is my pleasure to introduce Chanel Suda. Chanel received her Bachelor of Science from Virginia Tech, where she also she held internships at Stanford University, where she learned and mastered flow cytometry. In fact, everyone in the Pipkin lab, as well as on our floor, and particularly my lab, would run to her for any advice regarding flow cytometry, which is really a clear sign of a key aspect of a successful PhD student, becoming an expert in something, which Chanel has clearly established. Chanel joined the graduate program in 2016 and then ultimately joined the Pipkin lab. Briefly, the Pipkin lab is interested in how chromatin structure and transcription control the gene expression programs that establish and maintain the differentiated states of T cells. Chanel's work specifically looks at transcriptional regulation of CD8 T cell memory. All of her work has led to multiple abstract, poster, and short talk awards at several conferences, such as AAI, and further, she has won a Young Investigator Award from AAI and travel awards from NIH. Chanel's also participated extensively in teaching on campus, mentoring multiple summer interns, some of which have gone on to join other PhD programs, as well as ours, and has supervised master's students. To wrap up, she's published 5 co-author papers in journals such as Journal of Experimental Medicine, 3 other co-author papers in various stages of review in Molecular Cell, Immunity, and Nature, and has a first author manuscript published in Cold Spring Harbor, Perspectives in Biology. And I know she has a paper under review in Immunity, working on ETS1 and how it basically regulates TRM cells during infection. Her work and effort here will leave a lasting impression on Scripps and the immunological field overall, based off not only her science, but off her training in future scientists and their development as well. Job well done, and congratulations, Chanel.
Hi, it's my pleasure to introduce Michael Pigula. Mike came to Scripps from Boston College and joined my lab in our efforts to rewrite the central dogma and add new building blocks to the genetic code. And he made a major advance in that area when he figured out how to genetically encode cofactors as the side chains of amino acids to build catalytic sites, very simply. He then went on to apply the technology to the generation of safe live vaccines. Live pathogens are the best immunogens, but they're unsafe, so he figured out a way to make those pathogens conditionally dependent on an unnatural amino acid such that you could grow them outside of the host, immunize the host, and if they undergo several rounds of replication, they cease to replicate. And he showed those were terrific immunogens, safe, and couldn't escape. And finally started a project we're just moving forward in the lab, which was to ask whether we could rewrite a whole genome genetic code from a 3-base code to a 4-base code in the yeast mitochondria. Based on his amazing insights, productivity, and independence, he was hired over at Caliber Skaggs, and he continues to bridge basic research with the development of new medicine. So join me in thanking and congratulating Mike and wishing him the best of luck.
It is a pleasure to introduce Nathan Dow. Nathan is currently a senior scientist in discovery process. He is at Merck, actually at their London site, so he's come quite a ways to be with us today, about 5,500 miles that way. Looked it up on Google Maps. Nathan epitomizes the peripatetic scholar. He grew up in Vietnam, it's about 8,000 miles that way, and he attended a school for gifted children. He participated in national and international chemistry Olympiads, winning gold and silver medals. He then emigrated to Canada to attend University of Victoria, where awards and scholarships continued to rain down on him. My favorite is the Leo Neufeld Linear Algebra Award. So Nathan, I've brought my kids matrices for you to solve after the ceremony. Here at Scripps, Nathan published 4 first author papers, including a nice methodology in Science magazine and a short organometallic mechanistic study of his own design. But his main thesis work involved a platform to diversify and study the non-classical psychedelic salvinorin A. This is the psychoactive principle of Salvia divinorum, which has been cultivated for centuries by the Mazatec people of Oaxaca, Mexico, near the Sierra Mazateca Mountains. And they use it for ceremonial use. Recent studies by Johns Hopkins finds that salvinorin disrupts the default network of the brain, which is thought to underlie numerative, cognitive, and mood disorders, including resistant depression. So Nathan's work allows us to understand and perturb with atomic resolution the structure and function of this remarkable compound. As a result, we have some very exciting work with Leah's lab that continues to bear fruit even after Nathan's departure. We're very lucky to have Nathan's parents with us today, all the way from Vietnam, as well as his brother and partner, Aria. Nathan, long before you left the lab, you were no longer a trainee, but a peer, and I'm happy to make that official today. And I'm very happy to also introduce Eleanor Landwehr, who is currently senior scientist at Janssen Pharmaceuticals and joins us all the way from Maryfeld Row, about 2,700 feet that way. Eleanor entered my lab with the deck really stacked against her on 3 fronts. First, she graduated from the University of Wisconsin-Madison as a chemical engineering major, which is fine, Donna, which is fine. It's just not what our lab does. Second, she joined in 2020 during the COVID-19 shutdown. And third, actually, she was tasked with resurrecting a total shambles of a project that had just lost key personnel. So it's a testament to her tenacity and brilliance that she turned these lemons into probably the best lemonade you could imagine, in this case, her own Science magazine paper, a really beautiful publication. Eleanor's thesis involved the study of the neuroactive constituents of Galbulimima bark, which is used by the Gimi people of the eastern highlands of Papua New Guinea in medicine and in ritual. Because these compounds are used in traditional medicine, they already clear hurdles in modern Western medicine that are hard to reach, like oral bioavailability, that is, can you take it like a pill, and brain penetrance, that is, your body's natural barrier to keep it out of the brain. Due to Eleanor's thesis work, we're beginning to profile the targets of these compounds, some of which reduce muscle spasm, relieve fever, lower blood pressure, and affect mood and cognition. Now, only 2 protein targets has been identified, one by University of Sydney in 1988, and then another by our lab at Scripps in 2022. This latter metabolite exhibits highly pliable pharmacology and has shown robust efficacy in anxiety assays, which we could all use these days, I guess. We're now poised to discover numerous targets based on the robust platform that Eleanor helped to design. In addition to her work in the lab, Eleanor served as a teaching assistant, surf mentor, and lab manager, which was a major role and a huge help. Ellie, is an absolute tragedy that you're not in the lab anymore. You yourself are a walking antidepressant and a true leader, and I can't wait to see what you bring to science. I should also mention that both Nathan and Eleanor graduated from the PhD program in just 4 years. Major accomplishment.
It is my pleasure to introduce my student, Dr. Adriana Wilson. Adriana double majored in chemistry and biochemistry at Virginia Tech. After graduating from Virginia Tech, she worked at Pfizer and at the NIH as a post-baccalaureate fellow before joining us at Scripps. Despite her chemistry background, she took a chance and pursued a different research path, joining my lab to study a superfamily of transcription factors, nuclear receptors, in the immune system, specifically in Th17 cells. This ability to pursue any research discipline is what I believe one of the many strengths of our graduate program. Adriana basically uncovered an unknown role for the orphan nuclear receptor NR2F6 in Th17 cells, showing that it functioned as a critical early regulator acting to influence local chromatin states and gene transcription. Through this work, which was a Herculean task, and now we understand why it's an orphan receptor and many people don't work on it, she brought in and mastered a lot of techniques to the lab, all of which have definitely lightened my wallet substantially. However, it was worth it, and I think the story she has uncovered is very interesting, and hopefully the reviewers agree as well. During her tenure in my lab, Adriana was the recipient of several awards and fellowships, including the Scheller Graduate Student Fellowship, an NIH F31 Predoctoral Award through the NIDDK, various poster and travel awards through the AAI, as well as having several publications, one of which, as I mentioned, is currently submitted. And I want to conclude that it has been an honor and privilege to watch her grow into the independent scientist that she is today. She has been the go-to person that I have had in my lab, and now I have to figure out who the next go-to person is going to be in my lab, at which she's going to leave me bereft. I'm very proud to be here to watch her walk the stage and accept her degree. Congratulations, Adriana. A job well done.
Good morning, everybody. It's my privilege to introduce Dr. Chung-Che Liu, who arrived at Scripps from National Taiwan University, where he had obtained a master's degree from the Chan Lab, in which he had studied how sphingolipids may play a role in neurodegeneration. When I first met with Chung, he drew out his master's thesis with a twinkle in his eye, in which it had already become clear that he could tell the difference between a simple genetic study in which you remove a single gene and look at what happens versus being able to map an entire genetic pathway in which there are both positive and negative, agonistic as well as antagonistic forces at play. His main project was to characterize how a gene that was functioning in the gut was actually working in the brain, and to map out the entire genetic pathway by which it did so. Over the years, he displayed an incredible work ethic and a special type of intellectualism that's characterized by the finest molecular geneticists, in which they can go seamlessly from the abstract to the concrete and back to be able to really show us how something is working in a microscopic organism in which many things have to be inferred correctly. He had a lot of challenges along the way, but they were richly rewarded by a recognition that he received in the form of travel awards, as well as a Best Oral Presentation Award, as well as a Best Poster Award at international meetings ranging from Scotland to places in the United States. Not only was he recognized by the scientific community, he actually also had the honor of being featured on National Public Radio and National Public Television. So his dedication to his work was not just seen in the outside world, but felt very deeply by people within the lab. Not only was he a relentless force in doing his own work, but he also mentored undergraduates in the lab, two of whom are now going on to their own graduate studies in very highly recognized graduate programs. So as he plans his next steps in his career, I know that his dedication, his kind nature, and his extremely rich intellect will take him far in life. Congratulations.
My name is Andrew Su, and I have the pleasure of introducing Kai-Yu Chen. Kai-Yu earned her bachelor's degree in biology from National Chung Kung University in Taiwan, followed by a master's degree in bioengineering at UCLA. Kai-Yu honed her experimental skills at the Center of Biomedicine and Genetics at City of Hope, where she helped scale stem cell therapies for clinical trials. After some graduate work in computer science and machine learning at Georgia Tech, Kai-Yu joined Scripps Research specifically to pursue a doctorate in computational biology. Her thesis focused on the analysis of low-coverage whole genome sequencing data, which has the potential to be a very cost-effective strategy to conduct large-scale population-based genetic studies. Among her many contributions were the development of an efficient pipeline to impute genetic variation from these low-coverage sequencing data, the creation of low-coverage data simulation tool to aid the development of new algorithms, and then the development of her own algorithm based on deep neural networks. So in addition to developing an impressive skill set, Kai-Yu displayed an impressive degree of tenacity and perseverance during her PhD. Please join me in congratulating Dr. Kai-Yu Chen. I also have the pleasure of introducing Carolina Gonzalez Cavazos. Carolina's path here today began in her hometown of Monterrey, Mexico, where she also earned her bachelor's degree in genomic biotechnology at the Universidad Autónoma de Nuevo León. As an undergraduate and post-bac researcher, Carolina pursues a really impressive breadth of experimental research spanning genetics and cell biology applied to cancer and neuroscience. Upon joining Scripps in the fall of 2020, Carolina quickly pivoted from her experimental background and discovered the joys of computational biology. Her thesis work in my lab involved two major advances for AI-assisted drug discovery. First, she completed and characterized DrugMecDB, which is the first and largest database of manually curated drug mechanism pathways. This resource is critical for providing a gold standard benchmark for computational drug repositioning based on knowledge graphs. Secondly, Carolina developed her own algorithm called DBRX, a new method based on graph neural networks for generating drug repositioning hypotheses that really focus on the idea of providing clear mechanistic explainability. And in this work, Carolina displayed an exceptional ability to synthesize advances in this really fast-moving field and charting a path forward to new and novel contributions. So please join me in congratulating Dr. Carolina Gonzalez-Cavazos.
Double Dr. Khattab, I'm really pleased to stand before you and congratulate Ahmed on this major life achievement. Ahmed is an extremely strong and dynamic intellect. His work is centered on genetics and diabetes and spans techniques involving computational biology as well as implementation clinical science approaches. So his work involved the execution of a genome-wide association study, or a large scan, looking at genetic factors involved in diabetes complications. He developed a computational tool to enable genetic analysis for what is one of the largest and richest biomedical data sets in the United States, if not the world, and developed a machine learning tool for the genetically informed prediction of diabetes risk for screening purposes, including even economic impact analysis. Beyond that, he initiated an international collaboration in genetic studies, pretty much on his own, and even designed a clinical implementation study looking at the impact of genetic information in underserved populations. So really quite a breadth of different techniques, all focused on genetics. Beyond that, he's really just a warm and warm-hearted person. And I have no doubt he's going to go on to make a big impact in his community and improve the lives of people around him, both on a personal and professional level. I'll leave you with one word of caution. Don't bring a piñata around this man. He gets quite violent. Ahmed, although you attempted to batter my son, he has got a thick skull, so he's all right. He's like his father. I'm really proud to call you my prior PhD mentor, and really looking forward to continue to watch your son rise and illuminate the lives of others. Congrats.
Hello, I'm James Voss in the Department of Immunology. It's my pleasure to introduce to you today Mary Tenuta. Mary was one of the first people to join my lab after I became faculty at Scripps. So she helped not build only our program, our science program, but she helped to build our team and culture as well. The science developing genome-edited B cells to cure HIV was ambitious and unproven, but Mary was not afraid to break new trail. She helped recruit many of the students who followed her, and she made them feel welcome, supported, and excited to tackle big challenges. The culture she helped create says, we can do hard things, we're in this together, and no one defines our limits. Mary had to be a scientific pioneer from the start, and as many pioneers do, she took risks, worked outside of her comfort zone, and committed fully because she believed in the vision. Her hard work has resulted in many important contributions to our young but rapidly growing field. So specifically, she developed a number of precision genome editing methods to reprogram the antigen specificity of B lymphocytes from non-human primates. She engineered the cells to express a variety of HIV broadly neutralizing antibodies from humans and NHPs. And during COVID, when we had to be working on COVID or nothing, she engineered cells to express neutralizing SARS-CoV-2 antibodies. She spent a lot of cold winter weeks in Wisconsin, where she engineered cells, performed autologous transfer experiments and vaccinations to elicit transgenic antibody responses, and she is so far the only person to achieve that kind of a goal towards our goal of eliciting durable, broadly neutralizing antibodies as a HIV cure approach. Mary, I'm incredibly proud of how you, oh, sorry, I forgot. In order to not go to Wisconsin so often in the winter, she also developed an organoid model system where she could vaccinate engineered cells in secondary lymphoid tissues in vitro. So she's established that new system as well. She's published in Nature Communications and Nature Biotechnology and is under review at Nature Biomedical Engineering. Mary, I'm incredibly proud of how you've grown as a scientist and a leader. Your creativity, integrity, dedication, and personality will take you far. I can't wait to see what you accomplish in the future chapters of your research in immunology and gene therapy with Michelle Nussenswag at Rockefeller University in New York City.
I'm Assistant Professor Marko Mravich, and I'm here to introduce Dr. Colleen Maley. I'm going to start with some comments from her co-advisor, Professor Andrew Ward. It was an absolute pleasure to co-mentor Colleen and watch her develop as a scientist. She demonstrated tenacity, perseverance, ingenuity, and used a wide array of biophysical and structural tools at her disposal to tackle her thesis. She paved a lot of new ground, providing a solid foundation for targeting membrane proteins in a novel manner. Congrats on obtaining your PhD, and looking forward to working with you in the future. And for my comments on Colleen, she's the first person to join my lab, and it's been an honor to have mentored her. She used the opportunity of a completely empty lab space to paint on this empty canvas, creating a project to her liking, and applying our lab's technological experience into her interest in immunology, which I have very little experience, so much of the time Colleen was mentoring me. She blazed a path of firsts within our group, earning many personal awards and fellowships that reflect her incredible potential as a scientist. She started as a pure computational biologist and published the first manuscript coming out of my lab, which is a pure theory and simulation paper defining the mechanism of how antibodies can recognize lipid membrane components in HIV neutralization, revealing atomic details we couldn't see through experimental methods. One thing I did know about Colleen, when she came to Scripps, she didn't know how to use a micropipette, but she persevered, she learned, and ended her PhD developing a brand new experimental system in the lab, becoming a wet lab expert, and achieving one of the most difficult things to do in the protein sciences, which has led to a patent of a class of new, totally synthetic peptides targeting an important immune receptor we hope to develop as an anti-inflammatory. Now she graduates Scripps as a master, well actually a doctor, of protein design and engineering. She's, as us computer nerds would call, a full stack scientist, an interdisciplinary all-star. Very much looking forward to following their achievements over the course of your certainly bright career. Congratulations, Dr. Maley.
All right, I'm Andrew Ward in Disco Bio, and I have the pleasure of introducing Andre Nicolas Leon. So Andre is a proud Latinx Salvadorian citizen that grew up in the greater Los Angeles area, and his proud parents are joining us today. He did an undergraduate degree and got his bachelor's at Cal State Los Angeles. He also got a teaching credential from Loyola Marymount, and he actually taught high school before decided to do a summer internship here. And he really exemplifies how recruiting starts really early. And if we can engage these people and show them what can be done here, we can bring them in as grad students. And you've heard, a few of those examples today. Andre, this really bright, young, face when I gave my tenure talk in 2017, immediately came up after me and said, I want to do this with the technology you're developing. I said, that's 20 years in the future. Let's work on the next 5 years. I was lucky that he came into the grad program, joined my lab, ton of enthusiasm. I gave him sort of a blank slate in a sandbox, and then halfway through his PhD, we weren't making as much progress, so he had to pivot. And remarkably, went from what he was doing to really embracing influenza biology, understanding immune responses, and finishing on that note. So I will say, Andre has been an amazing citizen, starting up mentoring programs, mentoring and advocating for diversity and underrepresented minorities, also advocating for tenants' rights and doing things in the community, a real citizen scientist. I've provided the sort of guardrails for the last 5+ years. It's time to kick Andre out of the house, make him a little bit uncomfortable, but I feel really confident in doing that because I know he's going to do great things. And really, congratulations. It's been a pleasure, Andre. Always with a flair.
Good afternoon. My name is Jamie Williamson, and it's my pleasure to introduce to you Shiyu Dong. Shiyu, I first met Shiyu when she was an intern in the Rumsberg lab. We were very fortunate to recruit her to the graduate program, and I was very fortunate to recruit her to my lab. Now, she's had two major accomplishments here at Scripps. So the first was her thesis work on ribosome assembly. So she used cryo-electron microscopy to study the process of assembly of ribosomes in vitro, setting up a system where we could actually make a time-lapse movie of what was happening. And she could show that the order in which the domains assembled was the same order in which the ribosomal RNA was synthesized. Then she turned the problem on its head, and she changed the order of the RNA. So she made circular permutations such that the domains were made in a non-natural order. And remarkably, she showed that even though you synthesize the RNA in a different way, the domains assemble the same way. There's such tremendous pressure in evolution to do it this way. So she fundamentally wrote the map of how ribosomes assemble. And this is the stuff that's going to be in textbooks. Now, her second accomplishment, turns out our lab is right around the corner from Jinghuang Yu's lab. And this led to frequent and productive collisions with her husband, Jen. And so I can succinctly summarize her time at Scripps. In the Williamson lab, we do ribosomes and love. Congratulations.
All right, my name is still Luke Wiseman, and I'm a professor in the Molecular and Cellular Biology Department, and today I have the pleasure of introducing Nicole Madrazo. Nicole is a local San Diegan who began her scientific career as a high school intern working in my laboratory for 2 summers, where she demonstrated her early scientific excellence by earning authorship on a manuscript we published in Cell Reports. Nicole then went on to the University of California, Berkeley, where she received her Bachelor of Arts in Molecular and Cell Biology, while also pursuing research in an array of topics, including heavy metal resistance in bacteria, heart regeneration, and cell division in yeast. In 2020, she decided she didn't have enough of me yet, so she came back to Scripps to pursue her PhD in my lab. During her graduate training, Nicole primarily worked on projects focused on developing new strategies to activate protective stress signaling pathways to mitigate pathologies linked to neurodegenerative disease. These included work defining specific pathways activated by mitochondrial dysfunction in disease and developing new approaches to inhibit a process called ferroptosis that's involved in promoting neurodegeneration associated with many disorders. Through this effort, she published 2 first-author manuscripts describing her primary research in molecular biology of the cell and RSC chemical biology, as well as earning co-authorship in many additional manuscripts, including one published in ACS Chem Bio and another one that's in final preparation for submission. Today, Nicole is joined by her parents, extended family, and friends, who, if I know them, are going to hear from in a second. Please join me in congratulating Nicole on a fantastic graduate career.
Molecular cell biology. So it's my privilege to introduce Yujie Si. Yujie did her undergraduate studies at Peking University, where she worked with my good friend Xin Chen, the Dean of College of Chemistry. With Xin's strong recommendation, Yujie came to Scripps and spent a summer in my lab. I was so fortunate to recruit her to stay on. Yujie is such a smart and hardworking student. She always has her own ideas, even carrying them on, even when I told her no. So in the past several years, Yujie has contributed to several major projects. She was a key member of the team that developed FUCA-ID, a technique that enables the detection and enrichment of the full repertoire of tumor antigen-specific T cells from the tumor microenvironment. After getting this co-author paper in Cell, she went on as demonstrated for the first time that FUCA-ID can be used to isolate antigen-specific T cells, not only from solid tumor microenvironment, but also from the tumor-draining lymph nodes and peripheral blood. And remarkably, she also demonstrated for the first time that she can use this technique to isolate antigen-specific T cells from human cancer patients in collaboration with the GodKind lab at UCSD. Because the number of antigen-specific T cells isolated from tumors are quite limited, large-scale expansion is necessary for adaptive transfer. However, this process often resulted in T cell exhaustion. Yujie discovered that culturing T cells with a dietary sugar called sialic acid can uncouple proliferation from differentiation, allowing the generation of large number of stem-like T cells for patient infusion that can be possibly done in a clinic setting. So I would like to mention that Yuji's parents are here with us today, all the way from China, and her cousin and family members from Canada. Congratulations.
Hi everyone, I'm Xiaohua Wu from the Department of Molecular Cellular Biology. Today is really my great pleasure to introduce my student, Tong Wu. Tong completed his undergraduate studies at Wuhan University. In my lab, Tong took on a highly challenging project focused on the role of R-loop in DNA repair. So R-loops forms during transcription and occupies about 10% of the human genome. So they have important physiological roles, but unregulated R-loops are also harmful and a major source of genome instability. So Tong's projects aimed to understand how DNA damage response pathways function to repair R-loop-related DNA damage. Since R-loops are so widely spread, Tong's work has a huge impact on our understanding about the genome stability, cancer prevention, and even cancer treatment. So Tong demonstrates a strong sense of curiosity and eager to explore new approaches. He was the first one in the lab to try sgRNA library screening, and also the first one to establish high-throughput genome-wide translocation sequencing. He is also creative and highly independent. So the R-loop research direction is entirely new to my lab, and he pursued it largely on his own. So he established several new techniques essential for R-loop studies and designed new double-strand break repair reporters. So with all his strengths, I'm proud to say that Tong has truly met the challenge and did an excellent job on his projects. He has completed 2 manuscripts, cell reports and nucleic acid research, both currently under revision. So we look forward to seeing them in print in the near future. Congratulations, Tong.
Hi, I'm Xianglei Yang, professor in the Department of Integrative Structural and Computational Biology. It's my honor to introduce Dr. Ingrid Valley as she graduates with her PhD today. Ingrid grew up in Versailles, Paris, yes, 2 minutes away from that palace, and completed an elite 8-year Doctor of Pharmacy program at University of Paris, Dakar, earning 2 master's degrees along the way. Her passion for biomedical research took her from Lawrence Berkeley National Lab to Stanford to the Salk Institute before she joined the Scripps PhD program in 2019. In my lab, Ingrid investigated how mutations in the NARS gene, which encodes the arginine tRNA synthetase, can cause rare neurodevelopmental diseases through dominant negative mechanisms. And I would say Ingrid is the first person who worked on this protein in the lab. And her work led to multiple publications and presentations at international conferences. She was recently recognized with the Rory Bella Foundation and Young Investigator Draft Grants for her commitment to rare disease research. Beyond her science, Ingrid has been a leader, mentor, and source of energy in the community, championing women in science, mentoring a future PhD student so that he can work with her in the lab, and even organizing a post-pandemic lab running club. We're incredibly proud of her, and her father is here to join her, and we're excited to see her impact in the future. Congratulations, Ingrid.
Good morning, everyone. My name is Jing Kwan Yu from Chemistry Department. It's my great honor to introduce the first student, Tai Wei Chan. Tai Wei was born in Shenyang in the north part of China, and during his high school, he won national Olympic gold in chemistry, which then earned him a free ticket into Peking University. During his undergraduate, he spent 5 months in Stanford working with Barry Choate as an intern. And through Barry Choate's high recommendation, we were fortunate to recruit him to Scripps. So his first part of the thesis is about actually AI, how to use AI to basically beat us, in terms of design new catalysts and new ligand. And the conclusion is they cannot. That's the first part of his thesis. But I know it's a little bit disappointing. The paper is slightly disappointing, but not entirely to me, because at least I get to keep my job so far. So nevertheless, the study was able to show within the box, small box, where we designed a special chemical space, they were able to help us to tell us to improve in a small degree, but not get outside the box and tell us what to do better than what we invented. So that was the first part. As a result, that conclusion then prompted him to rush into the field in a huge rush in the last 2 years. He decided maybe it's better to still do experimentally. So fortunately, through the 2 years hard work, he was able to discover one reaction, that is to replace the CH bond, which is the lowest hierarchy in terms of oxidation state, to the highest one, which is a carboxylic acid, which hopefully will be very useful for drug discovery. With that, congratulations, Tai Wei, for your PhD. Also, it's my great honor to introduce the second student, Nick Chekshin. Nick was born in Moscow, and then he joined Oxford to do undergraduate. There, he learned really extensively computational chemistry. Basically, I would say he already almost has a PhD in computational chemistry before he joined the Scripps. But at the same time, in Oxford, he also acquired a pilot license, which he did not tell me until this morning. And I would think twice before I take him to my lab, accept him to my lab. But he told me that time, maybe I would point the door to Peter Schultz's lab. You could join Peter Schultz's lab for that. But nevertheless, since his arrival, he trained everyone in my lab, computational chemistry. I think it made a very positive impact in my lab, how to understand the mechanisms through computational study in collaboration with Donna as well in kinetic study. But I think the biggest contribution he made was the project connected to actually our beloved late Professor Richard Lerner. So the story was when I arrived the second day, Richard Lerner came to my office. He said, we just signed a big deal with Pfizer, and I promised them that you are going to introduce a fluorine, negative fluorine to do radio labeling through CH aviation. And the first time I heard that, I was like, before you promise, can you discuss with me first, Because that would be the last promise I would make in my promise. But nevertheless, we made tremendous progress in the basic fundamental understanding how to do fluorination for medicinal chemistry. I think we were one of the most used technology group in the pharmaceutical industry. But we can never do F- to do radio labeling directly on drug, because that's the perfect technology for PET imaging until, I would say, a couple months ago, in collaboration with BMS, and also with a tremendous help from another creative student for design a chiral ligand and chiral catalyst, we were able to do CH fluorination in drug molecule in 30 minutes. Because you have to do it in 30 minutes so that you can do the PET imaging with 2 drug molecules. And that work will be, hopefully the GADI we send back will come out in Nature Catalysis in next week. So that is a tremendous contribution to drug discovery. And actually, not only we did that, we also did that in selectively, so we can label the drug chirally, put a chiral fluorine on it at the same time with 99% EE. So with that, actually, he's going to join Caliber. He already joined the Caliber for drug discovery, so congratulations. It's my great honor to introduce my third student, Yi-Hao Li. So Yi-Hao was born in Tianjin, and then he went to one of the best chemistry school in Nankai University, and we were very fortunate to recruit him. As you can tell, he is by far the tallest student I've ever recruited. So in day one, I told him, unfortunately, I have extremely high expectation as well. But I didn't tell him why. So in the first 3 years, he was working on basically ketone aldehyde. As you know, if you are not familiar with CH aviation, let me tell you what is the problem with CH aviation. Very simple. CH aviation's most significant symptom is autism, because CH bond do not talk to anybody. So there's no way you can do anything with them, right? So he invented the first 3 years, he invented a new way to a catalytic reagent that will help ketone or aldehyde to talk to metal catalysts. And that was 3 JAKs in a row, back to back. I would say that's the best chemistry in aldehyde chemistry, CH aviation, by long, long history. But then I told him that's not good enough, because we don't want to even use a catalytic reagent to do it. We want directly to take a ketone. And then somehow you invent a chiral catalyst that would give the ketone substrate a dead kiss and give a deadly bite that would just bite off the CH bond at a better position selectively. That seems impossible, but a couple months ago, that paper came out in Nature. He's a single author that historically solved that problem. That just opened the chemical space, doubled the chemical space, because in the last 2 decades, we have worked on always acid amines that I would say we have done extremely well. But the other 50% of the chemical space for drug discovery is ketone and ester. And in that one paper, he showed you can really bite off CH bond from ketone ester with the Yi-Hao Li's magic catalyst, right? So with that, he is already recruited by David Minnan in Princeton without my permission. I didn't even write a letter. He told me, David Minnan called me, I said, I don't even know this. So congratulations.
So we made it. And just to underscore what you heard today, I mean, the graduate students are really the heart of Scripps Research, and everything we do here is due to their tireless efforts. And I should say, we're actually getting closer and closer to endowing every graduate student that we admit with a fellowship. And I want to thank all of you who participated in that. As people come onto the stage, I'll just finish with a lighthearted comment, which the faculty have heard before, but I'll tell you because you haven't. So Joe, Jack, and John wound up going to the doctor. The doctor had bad news for all three of them. You're all gonna die in the next 2 weeks. So they sat in the waiting room and they said, well, what are we gonna do? Why don't we get together at a bar tonight and figure out what we're gonna do with the last 2 weeks of our life? And so Jack got up there after a few beers and said, I figured it out, I'm going to Vegas. I'm gonna gamble, I'm gonna drink, and I'm gonna frolic with women, and it's gonna be the best time I've had in my life. And John got up there, what are you gonna do? I'm gonna go to Maui and sit on the beach and drink Mai Tais from sunrise to sunset. And it's gonna be the most serene 2 weeks of my life. They looked at the last person. What are you gonna do? Been doing my homework. Found a graduation ceremony every day for the next 2 weeks, and I'm gonna go to them, okay? And they look at him and they're like, why are you gonna do that? And he goes, it's gonna be the longest 2 weeks of my life, okay? So, on that note, I should say it also works for your mother-in-law too, okay? On that note, I will now confer the doctoral degree on the candidates. By the authority vested in me by the Board of Trustees of Scripps Research and on the recommendation of the faculty of the Skaggs Graduate School in Chemical and Biological Sciences, I hereby confer upon each of you the degree of Doctor of Philosophy. Graduates, please move your tassels to the left side of your mortar boards, and everybody, please join me in congratulating. So this concludes the conferral of degrees. The new degree recipients, their family and friends are invited to a reception right outside the Hazen Auditorium. And please, guests, remain in your seats while the ceremony concludes with a recessional. Thank you very much.